Hello everyone, welcome to Live Hope Fusion, where knowledge meets excellence. Myself, your educator, Dr. Saswat Kumar Mohanty, and in this video, I am going to talk with you about genetic mapping methods. And the first part of this genetic mapping methods is recombination frequency. So the recombination frequency is one of the most important part for genetic mapping. Okay, so because you when you find a genetic mapping, you have to find recombination frequency. So before starting of my class, I would like to announce that we are going to provide free webinar series for CSIR-NET licenses. So if you are interested, then you can find the Google form link in the description and fill out the form if you are interested. So coming to the recombination frequency. So, in genetic mapping is the mapping of two gene or three gene. So, why? In most of the genetic mapping, we used to find the whether these two gene are how much distance means it's not a same similar as physical mapping because in physical mapping you will to find the distance between two gene in the term of base pair. It can be a kilo base pair, it can be base pair. But here, it is based on the linkage and crossing over. Linkage and crossing over. So, because as you know, if two genes are far apart, means the chance of crossing over is more. If more number of crossing over, then there is a more number of recombinant offspring produced. Okay, if two genes are close together, means they are mostly linked. When the two genes are linked, the chance of the recombinant offspring produced is less. So the number of recombinant offspring produced will be less. So here, the recombination frequency or RF is called as recombination frequency. It denotes the frequency of recombinant offspring produced. Frequency of recombinant produced means we are just finding out the percentage of recombinants produced between two genes. If more number of recombinants produced means the two genes are far apart. If less, then the two genes are close together. So that's why this genetic mapping is also called your recombinant frequency, which is the percentage of recombinant offspring produced from the total number of offspring. See here, how to find? See when you have to do cross between two genes. Okay, where to which you want to find the distance or okay. So here when you cross, we'll get lots of offspring. Out of the lots of offspring, you have to find which of the offspring are recombinants. Okay, so that's why the formula for the recombination frequency will be the total number of recombinants offspring produced out of the total number of offspring in 200. It is simple. Just you are finding out what the percentage of recombinants offspring produced. So number of recombinants means where in this recombinant offspring the two genes having a crossing over. Then total number of offspring means out of all the number of offspring means number of recombinants plus number of non-recombinants. So here I'll go to show you about how to calculate the recombination frequency or RF. So here I gave you example on the eye color and wing type of Drosophila. Okay, so the gene for the eye color here I took was PR. The gene for eye color PR and VG is the gene for the wing type. So here PR plus is the dominant gene and VG plus is the recessive gene. As you can see in this picture, this red color chromosome is denotes the dominant chromosome, whereas the blue color chromosome is denotes the recessive chromosome. So how to find the recombination frequency? So in the first step, we have to do a cross between two homozygous traits. Two homozygous means one parent should be homozygous dominant. As you can see, this parent, both the chromosome is having both the dominant gene, means homozygous dominant gene, and other parent it should be a homozygous recessive gene. Are you getting it? So here, these two genes are PR plus and VG plus. These are what? These are your dominant gene, and here PR and VG, this is the recessive gene. Okay, so here what it took here? So both two parents, parent 1 is the dominant homozygous dominant parent, parent 2 is the homozygous recessive parent. So you know, if you cross between a homozygous dominant and a homozygous recessive, the offspring, the F1 hybrid will be a heterozygous. One chromosome will be a dominant chromosome and other chromosome will be a what? Your recessive chromosome. So here, as you can see, the red eye, okay, and the normal wing is the dominant character. So this is the red color, is the red eye, and dominant wing, this is the dominant character. So in the heterozygous, there are double heterozygous, because we are talking about two gene. In the two gene double heterozygous, we will find red eye and your normal wing. So now you got the F1. What is the second step? In the second step, we have to cross this F1 hybrid with a tester. Okay. So why we are taking a tester? See, now in the F1 hybrid, you are having a dominant character. Both the genes are dominant. Whereas the tester should be always a homozygous recessive person. Okay. Tester, you can say it as a test class. Okay, in the test class, we have to take a tester. This tester should be a homozygous recessive for both the gene. Why you are taking here a tester as a homozygous recessive? So, because when you are going to find your recombination frequency, you are not going to get offspring in the very less number. So mostly we have to cross them so that you should get around more too much offspring, around 500 or more than 1000 number of offspring. So after getting 1000 number of offspring, to check each and every one genotype is very difficult. So, in this case, when you do a cross with the tester, now your F1 hybrid is having both dominant gene and the tester is having all are recessive gene. Okay, so from the offspring, what offspring will get? The offspring will get, we can find the genotype, you can find the genotype of the offspring from the phenotype. Are you getting it? Means by looking at the particular offspring, particular individual, we can say the what is the genotype. Because if the dominant gene is present, means that is the dominant character. Is not there, means that this gene was not passed in the offspring. So if you find two gene are in the offspring, if two gene are having dominant character, means there was no crossing over. If in the offspring, if one gene is dominant and the second gene is recessive, means what? Here you will get one dominant gene here. This will dominant and one recessive for VG, means what? There was a crossing over. Are you getting it? That's why we are doing a test cross in this second step. Okay. So now come to third step. After doing the test class, you have to get the data from the F2 generation. Okay, in the F2 generation, we did, this is your F1 hybrid, double heterozygous, this is your tester, which is your homozygous recessive for both the gene. So, as you can see in the number of offspring produced. So those offspring, so both the dominant character of a particular both the gene. Okay, if a particular offspring, so dominant phenotype of the both the gene, your eye color and wing type, means what? It's the same as the what? F1 hybrid. If a particular offspring it showed both the gene having recessive gene, okay, means recessive character for both eye color and vestigial gene, mean that is exactly same as the tester. Means what? Here in the F2 hybrid, the parent 1 is your F1 hybrid and parent 2 is your tester. Okay, when parent 1 is F1 hybrid and parent 2 is tester, when you got both dominant and both recessive gene, means what? They're exactly same as their parent. So we can say that these are non-recombinant product. So in the non-recombinant product means they are the parental type, means exactly same as their parents. Then we have to count by what? By looking at the particular individual, looking at the particular Drosophila. From the phenotype, we deduce the genotype. So we can count how many number of this Drosophila so this genotype. How many number of Drosophila so this genotype. So here I have data here 1339 individual. So your phenotype as your parental 1 or genotype as parental parent 1 like what F1 hybrid, and 1195 they showed genotype as the parent 2 exactly same. So we can say these are the parental character and non recombinant. Then come to the recombinant product. In the recombinant product means what? One gene will be dominant and other gene will be recessive. As you can see here, this is recessive, here is dominant. Okay, so what? There was a crossing over between the eye color and the wing type. That's why one chromosome, one gene came from parent 1 and other gene came from parent 2. Here, this eye color gene came from parent 2 and the wing type gene came from parent 1. Means what? Here, crossing over occurs mean these are the recombinant product. So now we can calculate how many number of Drosophila showed these genotypes. So here I have a data 151 showed this genotype 1, recombinant 1, and 154 individuals showed this second recombinant type. So these are 151 and 154. These are the total number of recombinant offspring produced from the two gene. So next, this is the formula. Okay, here we have to use the formula for our recombination frequency. Okay, so as you saw here, the total number of recombinant offspring produced was 151 and 154. So I'll write here total number of recombinant offspring produced is 151 plus 154. Okay, so what is the total number of offspring? It will be what? 151 plus 154 plus what is that? Your 1339 and 1195. 1339 plus 1195. Okay, then multiply with 100. Okay, are you getting it? So see here 151 plus 154, that is your what? Total number of recombinant produced. So it will come around your 304. Sorry, 305 plus. And we are talking about total number of offspring. Here is not only recombinant. Total number of offspring means number of recombinant offspring plus number of what? Parental type. Okay, so this will come as your okay. So this is divided. Sorry, I admit this is divided by. Okay, so this is your 2839. Okay, into 100. Are you getting it? So here just we did what we did. We just calculate the total number of recombinant offspring produced divided by total number of offspring. Okay, and we divided it. So 305. Okay, 305 divided by 2839. We will get 0.1074 into 100. So equal to 10.74. Okay, so the recombination frequency. So the recombination frequency for this gene between your eye color gene and the wing type gene of Drosophila is 10.74. So how this recombination frequency is linked with what? Your genetic mapping. I'll show you here. So recombination frequency is directly proportional to the linkage mapping. Okay. So in this link, as I told you, this linkage mapping, it provides the estimation. Okay, what is the estimation of the two genes? They are close or far apart. Okay, and this is not the physical distance. So they told you larger the recombination frequency, the smaller the recombination frequency. So larger recombination combination frequency means two genes are far apart, whereas the smaller recombination frequency means two genes are close together. And the recombination frequency is directly proportional to the linkage mapping. And remember that the recombination frequency should not exceed 50%, because if it's more than 50%, then these two genes which you are finding your distance are very far apart. Okay, means they okay. So that is not a particular, not a feasible result. So the recombination frequency should be 0 to 50%, and the so if you get a 10.7 for what you call recombination frequency is equal to same as your, you can say 10.7 for map distance. So coming to the map distance, so the map distance recombination frequency, so the linkage mapping is also denoted by a what, like you are using base pair. So here this you have to use centimorgan. So 10.74 recombination frequency equal to 10.74 centimorgan. So the eye color and wing type gene of Drosophila are, you can say, they are 10.74 centimorgan apart. Okay, so this is, that's why, see, in the next class I am going to teach you about how to find the exact linkage mapping gene order of a particular, of two or more than two gene, which you get questions in CSIR. So here the example, through recombination frequency you can find out the distance and the order of genes. Okay, suppose you have to find the order of three genes, that is gene A, gene B and gene C. Okay, so that's why we need to find the recombination frequency between gene A to B, gene B to C, and gene A to C. So for all those things, the experimentally you have to do as I showed previously. So after you do the recombination frequency, you get the recombination frequency. So we can find out the order. So as you see here, gene A to B is 13.2%, gene B to C is 6.4%, gene A to C is 18.5%. So gene A to C is 18.5%. So gene A and C are far apart. Gene B and C are having 6.4 and B and A is 13.2, means B is the middle gene and B is the middle gene and it's close towards C. So the gene order is A, B and C. Hope you get it, and I like to get your comments how you feel it. And if you want to join for our pre-webinar series, kindly find the Google form link provided in the description. And this is the problem for you. I hope you can do this problem. Here I provide the recombination frequency of four gene, gene A, B, C, and D. Fill the comment box with your answer. Okay. Thank you everyone and have a good day.